In this section, we're going to focus a little bit more on the structure of the cell membrane, as well as talk about membrane transport, how things get in and out of cells. So the cell membrane itself, if you recall from our previous notes, it controls what enters and leaves the cell. It is semi-permeable. And it's composed of two layers of phospholipids, or sometimes this is called a phospholipid bilayer. Bi for two. And the phospholipids themselves, they consist of a phosphate, sometimes it's called the head, and these are called the tails. These are made of fatty acids. If you recall from our biochemistry section, that fats are nonpolar. They don't like water. They're hydrophobic. They hate water. And the phosphate part, it's polar, so it's hydrophilic. It likes water. And this is why it arranges in this bilayer, because the phosphates like to face the water, the watery environment, I should say, outside the cell as well as inside the cell. And since the lipids don't like water, they sort of face each other. And so I'll show you a picture. So this is the phosphate, and then these are your fatty acids. So the model is called the fluid mosaic model. It's phospholipids, but then there's proteins stuck in it. And as I mentioned, the phosphate heads are hydrophilic, and then the tails are hydrophobic. And so here's sort of a picture of how it aligns. So you can see here, these are the tails. Since they do not like water, if this was a cell, this is just a little section of the membrane. So you can imagine these are the phospholipids here, the bilayers here, going around. These don't like water, so they face each other. But the phosphates, they do like water. And then these are the proteins that are sort of stuck in the membrane, and they have multiple jobs. Some proteins carry things in and out. Some proteins, like this one, this is called a glycoprotein. This is like an ID marker. It identifies your cells as belonging to you. For example, your blood type identifies, you have blood type A or whatever, is these ID markers are responsible for things like that. So why is it called the fluid mosaic model? So a mosaic is one of those things where they have like tiles, and then sometimes they'll have like cement between them, and a lot of times it'll make a picture or something. It's called the fluid mosaic model because when scientists saw it, they thought of the proteins as being like the tiles, because they're all different kinds of proteins and they have all kinds of different jobs. And then the phospholipid portion would be sort of like the cement between them that sort of holds everything together. So that's where the name fluid mosaic model came from, is the fact that you have these phospholipids that are kind of fluid-like, and then in there you have these proteins stuck, and that's sort of like the tiles in a mosaic. So what do the proteins do? Why do we have those proteins stuck in there? Well, they have actually a lot of different jobs. I mentioned ID markers. So for example, your blood type is determined by specific proteins on your red blood cells that identify your blood type as being A positive or B negative. And there are other proteins that also, this is why you can't just get a kidney transplant from anybody. They're looking for somebody that's like a tissue match. This is these ID marker proteins that are on your cells that identify them, so that your immune cells do not attack and harm cells that belong to your body. They identify those cells as belonging there, and that way if they see cells that don't have the right ID markers, they know those don't belong, and that's how you can fight off viruses and bacteria and things like that. Transport proteins. These are going to carry things in and out of the cell, things that can't pass through the phospholipid bilayer, because the phospholipid bilayer is very, very picky. Very few things can pass through that. That's how the membrane can control what gets in and out. And so these transport proteins, you have some that only transport sugar. You have some that specifically transport sodium, etc. Enzymes. We talked about enzymes a little bit, actually. They were things that speed up reactions. So sometimes you have proteins in the membrane that just speed up reactions happening there at the membrane. Fibers. Remember, your cell has a cytoskeleton, and these fibers in the membrane can attach to the cytoskeleton to help your cells hold their shape. And there are some other things, but that just kind of gives you a general idea. Here's some pictures of some of these. So this is a transport protein that's carrying things in and out. Here's your phospholipid bilayer. This is an enzyme doing a reaction. This is called a receptor. So this is picking up a signal like from hormones, send signals to your cells that cause your cells to react. And this would be a receptor protein. This would be an ID marker protein. They have these little sugars sticking off of them that identify. This would be a protein that's helping two cells membranes stick together, and here's one that's attached to the cytoskeleton. So the proteins have all kinds of jobs, and you can see that they look kind of like tiles in that sea of phospholipids, and that's, again, why it's called the fluid mosaic model. Now, cells are in a liquidy environment, both internally and externally, and so substances need to get in and out. So just a quick reminder what a solution is. So a solution consists of two things, solute and solvent. In our cells and in our external cellular environment, water is the solvent. So you have water, a watery environment with stuff dissolved in it, salts, sugars, amino acids, all kinds of things. So just a quickie here. If a solution is 10% solute, it is 90% water. In other words, a solution would always add up to 100%. So you'd have a certain percentage of dissolved things, that's your solutes, and then the remainder would be water. This is going to be important for the next thing that we're going to talk about. So if a solution was 5% solute, what would be the percentage of water? Well, since they add up to 100, you're just going to do 100 minus 5, and you would have 95% water in that particular solution. So what does this have to do with anything? Well, things go in and out of cells, and typically they move from areas where they're more concentrated, higher concentration, to areas of lower concentration. So this is called diffusion. Sort of like when you spray cologne in the corner of the room, and then later, a few minutes later, everybody's like, I smell something. Somebody sprayed something. That's diffusion. That's molecules of cologne spreading out from where there's more to where there's less. And this happens across our cell membranes as well. And this is due to just random motion. This does not require an extra input of energy. This is just because of the energy that the molecules already have, just because of temperature. You heat molecules up, they move faster. So molecules are always moving just because of their thermal, just because they're warm. But they tend to bounce around randomly and end up moving from where there's more to where there's less. So this does not require energy from the cell specifically for this to happen. This is something that's happening naturally. And again, the natural direction that any particle that can pass through the membrane will go is going to be from where there's more of that particle to where there's less of that particle. And a good example of this would be oxygen and carbon dioxide. Oxygen is something you need. You breathe in oxygen, and carbon dioxide is a waste product. So oxygen constantly is going into your cells, and carbon dioxide is coming out. And it's going from an area where there's a lot more to where there's a lot less. And you might say, well, if oxygen's always going in, why is there less inside? Well, because we're going to learn in our next chapter on cell energy, because your cells are converting that oxygen into carbon dioxide. So you're using that oxygen up, so there's always less inside than outside, so it's constantly going in. You're making carbon dioxide in your cells, so that's building up, so it's going to tend to go the other way. It's going to tend to go out. And so molecules will tend to move from where there's more to where there's less until it gets to a point where it's kind of even on both sides. If you reach that, that's called equilibrium, and then they kind of go both directions at the same speed. So if you look at over time, if you added a molecule, let's just assume that this is a molecule that is able to pass through the membrane. It's going to start off high concentration here, lower here. Over time, it's going to tend to go inside. Now, does that mean that these can't go back out? No. It's random motion. So these could actually go back out as well. But think about it. What are the chances that this molecule is going to go back out? Since they're so spread out, there's a much higher chance of one of these going in than one of these going out. But eventually, when it's even on both sides, now the chances are equal of going in either direction. And therefore, you've reached what we would call equilibrium. So, suppose the particles of sugar can pass through this particular membrane in the picture. Draw an arrow showing which way it will go. So there are 4 different problems here. So let's look at our first one. So here's our first one. So we've got a lot inside, right? But we've only got 2 outside. Well, since the sugar would tend to go from where there's more to where there's less, assuming it can pass, the sugar would tend to go out. Let's look at our second one. Well, this time we have a lot more outside than inside, so we would assume the sugar would be more likely to go in until it reaches a point where it's sort of equal on both sides. Look at our next one. Now we have a 4% sugar solution and a 1% sugar solution. So we just said that sugar will tend to go from where there's more to where there's less, so the sugar would tend to go out. And in our last one, we have a 5% outside and a 2% inside. Sugar is going to tend to go from where there's more to where there's less, so the sugar would go in. Now, for all of these problems, we're assuming that whatever this membrane is, sugar is able to pass through it. If sugar was not able to pass through it, then that would be a different story, and we're going to talk about that in our next lecture. All right, and last but not least, equilibrium. Again, when both sides of the membrane have equal amounts, the cell reaches what's called equilibrium. Now, it is called net equilibrium. Sometimes it's also called dynamic equilibrium, meaning like dynamic, like it's in motion. And the reason why is because it's not that everything stops. It's not like, oh, there's 3 over here and there's 3 over here, so everything's even, let's just stop moving. The molecules are still moving. But isn't it true that if they're spread out equally on each side, the chance of one going this way is kind of the same as the chance of one going this way? So the concentrations, if this is, let's say, 5% sugar and this is also 5%, are probably not going to change. I kind of think of it as like Christmas time, if you remember, especially during COVID, and a store was really crowded, and like you could only have 100 people in the store. So you had this line of people outside that wanted to go in, and whenever somebody would come out, somebody else would go in. Well, wouldn't it be true that the identification of the 100 people in the store was constantly changing because you had different people going in and out, but then it was always 100 people in the store? That didn't change because every time somebody came out, somebody else came in. And that's what happens to a cell in equilibrium. The amount going one direction is equal to the amount going the other direction. And so that's diffusion for you. So tomorrow we're going to talk about a different process called osmosis, which is specifically the diffusion of water, because the truth is most molecules can't pass through the membrane, but water can. So this is going to change things. What's going to happen if you have a bunch of sugar over here? Remember, this is a solution, so this has got sugar, but the sugar is in water, but the sugar can't pass. But water can pass. What's going to happen? You're going to have water, sugar, and water on this side. You have only water on this side. If the sugar can't go anywhere, what's going to happen? And so that's what we're going to talk a little bit about tomorrow.